Okay, hi everyone. Happy Friday to you. Um, let's continue our discussion about antibodies and the B cells that make antibodies. We looked at this last time when we introduced the structure of an immunoglobulin. Remember, immunoglobulins are these special molecules, these proteins that are made by B cells. And the purpose of an immunoglobulin is that it allows your body, your immune system, to produce many, many different shaped immunoglobulins with the hope that some of them, by chance, will be the right shape in order to be able to detect or bind to the surface of some sort of molecule coming from, let's say, a pathogen, either directly on the surface of that pathogen, or maybe it's a molecule that's made by the pathogen. So it serves as evidence that there's an infection inside of your body. And importantly, remember that this would be a pathogen that maybe you previously have not encountered before. And it is something that your innate immune system, that you come pre-programmed with the knowledge of throughout the course of evolution, right? Your innate effectors aren't able to properly identify it. So things like complement proteins don't attach well to it by themselves. Macrophages, maybe neutrophils, do a very poor job of recognizing and phagocytosing this bacteria, for example, or virus. And therefore, your body needs to come up with a new way of detecting this thing. And valuable time has gone by during which your innate system has struggled to kind of keep up, but it's failing to do so. The bacteria or the virus is really having an opportunity now to spread in your tissues, right? So this is bad clown or exotic clown now making his home in your body, and you are getting really sick, right? So that at that point, it's worth it for your immune system to really wake up the adaptive immune response. And one of the first things that has to happen then is that your immune system has to learn to actually recognize that bad clown really is a threat. And he is what your immune system is looking out for, right? To try to identify him and destroy him before you continuously continue down the path of getting so sick that you actually end up dying. So we said that the immunoglobulins then represent the sort of wanted poster, the poster that has to first know the picture of bad clown, or in this case, what we're saying is these are immunoglobulin molecules that have to be able to successfully bind to some feature of the pathogen. So some particular antigen that's present on the pathogen, right? Remember, an antigen is the actual molecule that your immunoglobulins are physically recognizing. And even then, within that antigen itself, you know, an antigen could be a pretty big molecule. There'd be many sort of three-dimensional surface features on it. A particular immunoglobulin would only be able to recognize, you know, one little aspect or one little three-dimensional feature on this molecule. Right? And that actual surface is known as the epitope of the antigen, right? And the antigen is from the pathogen. So oftentimes, when you hear about things like, you have a certain antigen that's present, right? That is the molecule that's coming from, let's say, the bacteria, or is on the surface of the bacteria, right? And that molecule is the key molecule that your immune system can easily target and recognize, and it does so by making immunoglobulins that recognize specific little epitope features of that antigen. Last time we showed an example, if you have a puzzle piece, you can have multiple antibodies, each recognizing different epitopes of the little puzzle piece. And importantly, we said that the example of the jigsaw puzzle, when I shook it up and was able to find a couple of random pieces that had just come together by chance, right? They were able to stick together just by having close enough fit, right? They are not the puzzle pieces that are meant to go together, but close enough fit at least allows them to kind of stick together a little bit, right? And that's the concept here. Your body is going to produce a wide assortment of different immunoglobulins, some of which hopefully will be close enough fit to be able to attach to a novel antigen from a novel pathogen and start your adaptive immune response being able to kick in and hopefully save the day. Right? So we said that these immunoglobulins are these Y-shaped molecules. Remember, they are proteins. They're made of 4 peptide chains, 2 heavy chains. So in here, the blue is 1 heavy chain, the yellow is another heavy chain, and then 2 light chains, the red ones. Each arm of this Y-shaped molecule, at the end, we have what we call a variable domain, right? So this is the part that will vary in shape from immunoglobulin to immunoglobulin. And we looked at last time that if we were to zoom in at the variable domain, in this case, just the variable domain of, let's say, the light chain, okay, up here, right? It is 1 peptide strand, so 1 chain of amino acids that folds up into this globular domain. And importantly, most of it is pretty conserved. From immunoglobulin molecule to immunoglobulin molecule, most of this kind of yellow part in this box on the right here would be the same amino acid sequence. But you would have these short stretches, these 3 short stretches where the amino acid sequence is widely variable from one immunoglobulin molecule that one B cell knows how to make compared to the immunoglobulin molecule on the surface of a different B cell, right? And that's because these 3 loops really help form the 3-dimensional surface that hopefully kind of matches up with the 3-dimensional surface of the antigen, right? To get close enough fit to start things off. And remember that each arm is actually not just the 3 variable loops of the light chain, but also there's the 3 variable loops of the heavy chain. So each antigen binding surface is actually 6 of these loops together. So to help illustrate this, you know, oftentimes I think students lose track of the fact that when we look at these globular structures, right, we tend to forget, oh, they're a protein, so therefore they're actually made by a peptide chain of amino acids that have to fold up. So I have here, you know, this is like one of my kids' fidget kind of toys. You know, it's meant to be for like kids who have like ADHD or something. So that's why my kids don't need it anymore, but I play with them all the time, right? Anyhow, so we have a chain of amino acids, right? Let's say, so this is 1 peptide chain of amino acids that's coded for by a gene, right? That then gets transcribed and edited into mRNA and then translated into a chain of amino acids. So let's say this is the heavy chain, right? So the end of the heavy chain folds up into a globular structure, right? That's what we see on the screen here. That would be like this part here. The rest of the heavy chain kind of continues off. Maybe I should hold it the same way that it shows up in the picture. I guess it's like that. No, it's backwards. Okay. Right. And that would then form the rest of the Fc region of the antibody, or the stem. Right. Then you have the light chain, which is again a peptide chain of amino acids that's coded for by a light chain gene somewhere, and we'll look at that, right? And so you end up making the light chain. And so it's the combination then of the light chain sitting on top of the heavy chain. This surface over here then is the unique 3-dimensional surface that's formed because the light chain and the heavy chain have these variable loops. You get a unique 3-dimensional shape. Right? And then along comes one day, right, you're infected by bad clown. And bad clown, either on the surface of bad clown, or maybe this is something that bad clown releases, are small little molecules, right, that might serve as a unique 3-dimensional signature that bad clown has. All right, so keep in mind, this might be something that is on the surface of a virus particle, a virus capsule. This might be on the surface of a bacteria, or this might be something that's released by the bacteria. Right? And your body's having trouble beating bad clown with just your innate immune system, so you're getting sick, right? So fortunately, you have these B cells that are being made that are making these immunoglobulins. Now remember, of course, this is just one arm of the immunoglobulin. If you look at the slide, right, the other side, it's kind of like the exact same thing, the heavy chain and light chain, and that's how you get the 2 arms. But let's just look at one arm, right? So the 3-dimensional shape then, right, by chance, just like the two random puzzle pieces in the box, is the right shape then to be able to bind to this antigen. Right? And the actual features of the antigen that it's binding to would be the epitope part of the antigen, whether this is free floating by itself or attached to the surface of bacteria, right? In which case this antibody or this immunoglobulin would be able to bind to the surface of the bacteria. So B cells that are making these immunoglobulins, they contain the information to be able to actually do this process of making unique immunoglobulin structures. We introduced this last time as saying that immunoglobulins are first expressed as proteins, these Y-shaped proteins that are just anchored to the surface of the B cell. In that stage, when they're still anchored to the surface, they're basically acting as receptors for the cell. So we can also just call them B cell receptors, right? So if you read somewhere that they're talking about B cell receptors, you realize, oh, these are the immunoglobulins when they're still anchored to the surface of the B cell. At this point, this B cell with these B cell receptors, it has very little interest in releasing these as free floating immunoglobulins, right? Why? Because at that point, this B cell that, let's say, just finished being formed in your bone marrow and now is released in your blood is just one of several 100 million B cells that your body will make today, each one with a unique immunoglobulin on its surface. In other words, this one B cell in the cartoon, right, is covered with these B cell receptors. Each one of these immunoglobulins is identical because that is the version of immunoglobulin and its variable three-dimensional shape that this particular B cell knows how to make. Right? Another B cell that's born at the same time, that's coming out of the bone marrow, it would also be covered in immunoglobulins, but those immunoglobulins would have slightly different antigen binding domain shapes on them. Okay. So if it's just going to be one of a few 100 million, it's like the puzzle pieces in the jigsaw puzzle box, right? Most of them, right, are not going to be the ones that end up matching up close enough fit with anything, right? And so at that point, all of these B cells, none of them are any more valuable than any other that's being made. So that all changes, though, if the immunoglobulin on this B cell successfully binds to something, right? In this case, the antigen that's on the surface of this bacteria, then that B cell suddenly, right, it's pretty valuable now because its unique immunoglobulin has just proven itself to be very useful, right? And this B cell is then chosen because it knows how to make this particular immunoglobulin shape that has suddenly proven to be very valuable, because this is going to be the start of how your adaptive immune system is going to spread the word that this is a threat, right? That's bacteria. And we already discussed, even though we'll go into much more detail as we continue with our lectures, that this B cell may be then told, okay, at this point, start making the free floating soluble version of your immunoglobulins that we call antibodies, right? So the B cell will turn into plasma cells, and then they will start making tons and tons of the antibody form of the immunoglobulin that can then go and start covering and opsonizing things like the bacteria. So here's the question. From that little story that I just told you, right, already you get a picture about why it's important that these B cells have to be chosen, right, in order to go through the trouble of making antibodies. Because if you're making hundreds of millions of them every day, only a teeny tiny fraction of them actually make immunoglobulin shapes that are relevant to the particular pathogen that you are now experiencing an infection from, right? All the rest are pretty much useless, okay? So you might say, well, but then wouldn't it just be better if the B cells, to be safe, just automatically make free-floating antibody versions? So your whole body, your whole system would be filled with a multitude of different antibodies, and then some of them, by chance, would be the close enough puzzle piece that binds to the surface of the bacteria. You might say, that makes more sense to me, because then you know you have a bigger chance of the bacteria being covered. But that's not how it works. And there's an important reason why. Right? Why does the Ig remain as a surface receptor until this process of being chosen happens? Right? To illustrate this, here's kind of a analogy, right? For those of you who take or who have taken Bio 275 with me, I'm very fond of this idea of proteins that are made by our cells being thought of as like cookies, right? And that in order to make a cookie, you need a cookie recipe. And the cookie recipe contains all the information you need to actually make the final result, the product of the cookie. And of course, the cookie itself is quite different than the piece of paper that has the recipe on it, right? Because the cookie itself is what you can actually enjoy eating. Just like the protein itself is the thing that actually carries out the function of what its gene is encoding for or intending it to do, right? And the gene encodes for the sequence of amino acids that makes that protein. So why then does an Ig protein on the surface of a B cell, if the B cell already knows how to make it because it's coming from the B cell's own DNA, right, that gets transcribed and then translated to make the protein, then great. Why doesn't the B cell just make a whole bunch of free-floating versions of this? It's because, just with the cookie story, hear me out here. Let's pretend, right, that there's a big mainland cookie company that has various stores, in malls all over the mainland, and they want to start opening business here in Hawaii, right? And so they figure in order to kind of drum up some hype so people know about, this new cookie store, they decide to have a cookie contest. They figure, we're opening in Hawaii, so maybe our signature cookie should be like a white chocolate macadamia nut cookie, right? Because that's kind of how mainlanders think about Hawaii, right? But anyhow, so they post this contest, and you see flyers around the UH Manoa campus that says, there's this contest, come up with your best white chocolate chip mac nut cookie, and then we're going to judge them. And the winner of this contest will get like a $1 million contract, right, to buy their recipe and have them perfect the recipe as the signature recipe for this cookie store. So on the contest day, let's say there are like, 200 students who show up with plates full of their cookie. Right? And I should say, because this needs to be a cookie that will kind of work for this company, it has to kind of be a particular shape, a particular size, right, in order for it to be consistent with the cookies, with the company's other cookies that are in like the display case and stuff like that. So there are certain rules, certain limitations or parameters of what your cookie might look like. As a result, the students show up with their plates full of cookies. There's two ways that the judging could happen. It could be that the judges are just standing at the door, right? And as people are filing in, they just say, okay, go over to that table over there. You'll see a whole bunch of blank or empty plates there. Just put like 5 of your cookies on that plate, all right? And then have a seat, okay? And so then now you have all these plates of cookies, right? And the judges begin to taste them. And the judges finally come down with like their 3 favorite ones, 1, 2, and 3, right? And they hold up the cookies and they're like, whose cookies are these, right? Guess what, right? For $1 million at stake, like, the whole class or the whole audience raises their hand. That's my cookie. That's my cookie, right? So that's the problem. Because you've disconnected the cookie from the student who actually knows the recipe from the cookie, you've now lost the ability to actually identify how to make more of this particular cookie, right? The other way it could be judged is that one by one, the judges sit up at the table and they call the students up. Okay, bring me your cookies now. And the student brings his or her cookies and the judges taste it right there, right? And they know that this cookie is coming from this student right in front of them. Then when they're all done scoring it, right, they know who has the recipe, the unique recipe that made that cookie. Okay, not just the single winner, but they might have a second place and a third place, right? Because those other cookies, right, there is some possibility that they can be tweaked in a way, right, to be just as good, if not better, than the first cookie. All right, so, right, that's what we're talking about here. The immunoglobulins, the cookies, right, are based upon unique recipes that only that B cell has come up with, right, to make that immunoglobulin. So you need to keep that immunoglobulin on the surface of that B cell. So when it is finally chosen, because in like the second slide of the cartoon here, right, and the bacteria actually has an antigen on it that this immunoglobulin recognizes, right away you know what B cell it is that possesses the recipe for this immunoglobulin. All right, so how then could it be that these B cells each have a unique recipe for an immunoglobulin, right? The shape of the immunoglobulin. Because after all the B cells in your body that are being made right now as you're sitting here, and today hundreds of millions of new ones will come out of your bone marrow, right? All of them come from your genome, right? They all start off with the same stem cell in the bone marrow that then divides and differentiates, okay? And yet, how can you get hundreds of millions of variations of proteins? Now, for those of you, again, who have taken cell biology, you know that through the process of transcribing DNA to make primary RNA transcript, that primary RNA transcript can be edited somewhat, right? Exons can be kept, introns can be cut out. But even then, so that then results in a different messenger RNA that can then be translated into a protein. So you might get a few variations of one protein from one gene, right? But the number of variations you can get is pretty limited. Typically, I think we now understand that a gene in our body, on average, can make maybe like 3 slight variations of a protein based upon RNA editing, right? So that mechanism cannot explain being able to have hundreds of millions of different B cells with hundreds of millions of different unique Ig shapes. So it's got to be more complicated than that. And so this mechanism is a fascinating mechanism. I don't think, chances are you have not heard of this before. The first time I learned about this, it really blew my mind, right? It happens through a process known as VDJ recombination, right? That's the first step of this. The second step happens through a mechanism known as junctional diversity. And then the third step just has to do with combining light chains that you make with different heavy chains that you make to get different variations of the finished Y-shaped molecule. So let's start in with this VDJ recombination, because this probably is the most tricky part of understanding it. Right. So the immunoglobulin peptide chains, these proteins, are expressed by 3 different gene loci, or 3 different gene regions on different chromosomes in our body. So remember the structure of the Y-shaped immunoglobulin. You have the 2 heavy chains. So these are the big ones that start from the top here. Remember, it's one continuous chain that's all wrapped up in these blobs, just like my little ADHD toy here. Right? The heavy chains go from the top all the way to the bottom, all the way from the variable region where it contributes to half of the antigen binding domain, the unique part that's different from Ig to Ig, all the way through the more constant parts that are sort of standardized, right, down to the end of the stem, or the end of the Fc region that attaches to something like the B cell. Then, so you have 2 of those, then you have 2 light chains. Okay. So the heavy chain is just the heavy chain. We have one gene or one group of genes in one location on chromosome 14 that makes our Ig heavy chain. Right? The light chains, we actually have 2 different genes. We have a lambda light chain on chromosome 22 and a kappa light chain on chromosome 2. So these lambda versus kappa light chains are somewhat interchangeable. In other words, if you were to look at one immunoglobulin molecule that you've sort of isolated and you're studying it, it would either have a pair of lambda chains or a pair of kappa chains. 33% of all the immunoglobulin molecules floating around your body, they use the lambda chain version of the light chains. The other 66%, the majority of immunoglobulins in your body, would have kappa light chains. We still don't know the difference because at the end of the day, when it's all put together, they function exactly the same. Evolutionarily, there probably is a reason why we have kappa and lambda chains. It may just be to increase the success of our B cells in actually being able to express these light chains. So maybe that's why we evolved with slightly different versions of it. Anyhow, this illustration on the bottom then shows you these locations. So on chromosome 22, so let's look at this top one here. This is a stretch of chromosome 22. Remember, a chromosome is just one continuous long DNA molecule, that's normally wrapped around things like nucleosomes and things like that to be present as chromatin. But if you unravel it, one chromosome is just one long strand of DNA. So on chromosome 22, there's a particular spot where you have a group of these gene fragments. Right? Each one of these represent a coding sequence that can be transcribed and translated, right? But each one by itself is just a piece of a gene. Right? You have a large number of these different red blocks, which we call the variable blocks. Right? Then you have a more limited number of these yellow blocks, which we call the J blocks. Right? So these are the joining blocks. And then you have several of these blue constant blocks. All right. Just looking at this top one here and looking at the heavy chain, one of these light chains here, you can kind of see the color, right? So the red variable region of the chain is going to be mainly based on these red variable blocks, okay? The reality is the finished variable region also has the yellow block in it as well. The blue blocks here, the constant blocks, they're the ones who make the constant region of the chain. So in this top one here, it's almost like you see it stretched out, and each one of these different fragments is an opportunity. Right? It is a choice that your B cell can choose from. And what the B cell will do is it will pick one red block, one yellow block, and one blue block to put together to make out of these separate fragments one complete gene that then, when that is transcribed and translated, gives you one of these chains, all right, in this case, the light chain. So the lambda and the kappa light chains are very similarly structured. So again, kappa is a different light chain gene locus. It's on chromosome 2, but it also has a number of these variable red blocks, a few yellow J blocks, and a few K blocks, okay? Not K blocks, but C blocks, all right? So notice that in each of these cases, you have like 30 or 35 different possible fragments or pieces to choose from, right? Maybe 1 to 5 different yellow blocks to choose from. Right? The heavy chain adds a different type of block in here, the D block. These are these green ones. So in the heavy chain locus, you can choose one of these 40 different red block fragments, the variable ones, choose one of these 23 D blocks, right? You have 6 different J blocks to choose from, and then your constant blocks. So one way of thinking about this is these are all slightly different ingredients that are part of this recipe that's going to be put together. Right? And these different groups, the reds, the greens, and the yellows, they're all different sort of like main ingredients that need to go into the cookie recipe, like flour or sugar. Right? But when you talk about flour, there are many different brands of flour that you could use, right? Like coarse, general flour, bread flour, whatever, right? Or sugar, right? There's many different brands of sugar that you could use, right? And so you have many choices, and your choice of different ingredients that you make makes for a unique finished recipe that, once translated, creates a unique protein product. Right. So one thing I want to show you on these cartoons, well, a couple of things I want to point out. One is kind of obvious, but it's easy to forget about this, right? These are drawn as these colorful blocks, as though somehow, if you were looking at the DNA, you could actually tell that this is a V block or a J block or a C block. In reality, you cannot, right? These colors, these cartoons are just meant to symbolize different stretches of DNA, when in reality, if you're looking at the DNA, it's just all A's, T's, C's, G's, different combinations of those, right? But functionally, different regions of them are these gene fragments. And your B cell, as it's developing in your bone marrow, right, has to be able to recognize that these are actually different gene fragments, and importantly, somehow be able to choose one of each kind, put them together so that you get one continuous finished gene. Right? When I was a student, I had never heard of anything like that before, right? So I thought it was crazy because it kind of went against everything I understood about how our cells use our genome, right? Because we're talking about like editing the genome here, right, to come up with different combinations. So how do these do that? How do the cells do that? These different blocks, right, are surrounded by DNA sequences that are highly conserved. We call these recombination signal sequences, or RSSs. One type of RSS is made of 7 base pairs of DNA, a particular sequence of 7 base pairs of DNA that's highly conserved in evolution, right? And you find these, right, surrounding these different blocks in that stretch of the chromosome. So because they're made up of 7 base pairs of DNA, we can just call it a heptamer, right? That's just a generic way of saying a little stretch of DNA made out of 7 base pairs. Another similarly conserved sequence is made up of 9 base pairs. So we call that a nonomer, right, meaning 9. Right? And so these highly conserved sequences of 7 and 9 appear close to each other, spaced apart by either 12 base pairs of random DNA that doesn't matter what the sequence is and it varies from spot to spot, or 23 base pairs of DNA that are, again, highly variable and it doesn't matter. So if you look down here, you can see this here. In the lambda chain, what they've done is they've zoomed in on just one of these red blocks, right? So keep in mind, there might be like 30 different of these red blocks to choose from, right? And then somewhere way down the strand, you start looking at the yellow J blocks, right? And there's maybe like a dozen or so yellow J blocks to look for, to choose from. Keep in mind, you see how they broke the strand here by putting dots, right? This is just to remind you that these are not like right next to each other in real life. There could be thousands and thousands of base pairs of DNA in between them. But for our discussion of what's going to happen here, we're focused on these 2 spots, this particular red block that was chosen and this particular yellow block that was chosen out of the possible choices. You can see next to the red block, you would find that highly conserved heptamer DNA sequence, then 23 random base pairs of DNA that could be anything, then that highly conserved nonomer, 9 base pairs. Upstream of the chosen J block, you have the heptamer here, then 12 base pairs of random DNA, and then the nonomer. Right? So what this is doing here is it is actually identifying, it's like a little flag that indicates that this DNA sequence actually is a red block or a yellow block. And importantly, that particular 7-23-9 and 9-12-7 acts as a key, right? A key that allows you, or the cell, right, to actually start moving these blocks together and keeping the orientation of the blocks correct. Because once these blocks are together and the sequence of both of them is meant to be read as a DNA sequence that's going to be transcribed, you better make sure that they are attached in the right orientation, because if one's flipped around backwards, then the sequence becomes meaningless. Right? Here you can see the same concept in the K chain and in the heavy chain. The heavy chain, again, has that extra green block, the D block, right? But you can see the D block is flanked on either side, 7-12-9 going off to the left, 7-12-9 going off to the right. So this is what we call the 12-23 rule. In other words, one of these sites that has a 23 base pair spacer in it is only allowed to pair up with another site that has a 12 base pair spacer in it. Okay, and that's how the cell manages to keep everything oriented correctly as it's joining these together. So there is a big protein complex made up of several enzymes known as the VDJ recombinase complex that will actually take care of choosing these things and bringing them together. So that's what's shown here. So let's look on the left over here. This is what we're looking at, right? So from left to right, we were saying there's a red block, a V block, the 723.9 RSS sequence that's downstream of it. Then some distance away, because there's these dotted lines here, you have another one of these RSS sequences, this 912.7, that leads into this yellow J block. So imagine that this is that strand of DNA here, going from left to the right, and then it's bending over and continuing on, right? So here's the V block with the 723.9 next to it, and then downstream somewhere leads into the 912.7 that leads into the J block. Okay. This green thing with these two ovals represents the big recombinase complex, right, or RAG protein complex that proceeds to recognize the RSS sequences. You can see the ovals, it's sort of showing that the ovals are somehow recognizing those consensus sequences. And there's other parts of this protein complex that take care then of joining together these blocks so that this V will then be connected with this D over here. So here we see it illustrated again. You have a strand of these blocks, right? And then you have this big RAG protein complex. You can see here at first it's attached to the recombinase sequence or the RSS sequence that's right in front of this J block. Again, this is just one yellow J block out of potentially what, it's like 6 to 8 different ones to choose from. So by chance, randomly, the protein complex, the RAG, has attached here and chosen this one, right? Then it'll bring it all the way around to attach to one of these V blocks here. In this case, look what's happened. It's actually skipped over the V2 block and decided to attach to the V1 block on this side. Again, because the V1 block has an RSS sequence, in this case, maybe this is a 723.9 or something like that. And you can see over here, the RAG complex then loops this around, right? Bringing the chosen V block next to the chosen J block. And look what happens. The complex actually cuts the DNA right there. So this loop of DNA, right, that is part of your genome, right? This is part of the sequence that you were born with, that you got from mom and dad, suddenly is cut loose. And it just gets recycled. It is destroyed, right? The cell no longer holds on to it. And whatever colored blocks happen to be in this part that was skipped over, for example, this V2 block, it is lost in that cell. That cell no longer has that as part of its genome anymore. The RAG complex then needs to join up the two cut free ends of the DNA here. And once it's joined, you now have successfully, right, a V block next to a J block, right? And this is the part that's then just going to float away and be degraded and eliminated. Right. So I think that's a really confusing kind of concept. So to illustrate that, I have a little demo here. Let me stop this sharing. Okay. So this length of rope represents one of those chromosomes. So it's chromosome 14 because it actually has these green D blocks in there as well. Right, so I have on this rope this long length of DNA that represents your chromosome. Right, you have multiple red V blocks to choose from. These are the variable blocks. I have 3 of them, right, starting from the end over here. Then I have a couple of green D blocks, and then you keep going down because this is meant to show you these things are actually physically very far apart from each other on the chromosome. Then we have some yellow blocks. Here's a yellow block here. Right. And eventually the blue constant blocks, right? All on the same strand of DNA because this is all chromosome 14 or part of chromosome 14. We're zoomed in here. So the RAG protein is going to recognize these recombination sequences. So each one of these, for example, this red block here, right? I guess I should hold it this way so it looks more like it's in the cartoons, right? The red block will have downstream of it a RSS sequence. So let's say this is a 7, and then there's a highly conserved 7 DNA base pairs that you see over and over again, you know, in different places. There's one over here too, right? Then you have a purple nonomer, so it's 9 DNA bases that you also see in other places like down here, so it's highly conserved. But the part that's in between them, right, it's not important what this is. It just so happens there's 23 base pairs of DNA in there, right? So that acts as a key. So the RAG protein complex is like this oven mitt, right? I know this is like really kind of ghetto, but you know, it's my whole problem with the state school budget for props. Anyhow, right? So this protein complex is made of different proteins that together give it a unique shape, right? This oven mitt shape. Part of this protein is the right shape to recognize DNA that has this nonomer or protein 9 DNA base pair sequence, right? Another part of this protein, the orange part, is the right shape to recognize the heptamer sequence, right? So this big protein complex will randomly choose one of these red V blocks out of, let's say, 30 choices, and it'll settle in so that it grabs the orange part, can grab the orange part, the purple part can grab the purple part, right? And then what it proceeds to do is elsewhere on the gene, on the chromosome, let's say there are these green blocks, right? These are, let's say, the D blocks. Right? They also have those same sequences, but of course the spacer between them is different. So that's because the oven mitt, if you look at it, it's not symmetrical, right? So if it binds this one and this one here, in order for it to successfully also bind the RSS sequence surrounding the green one here, if you were to try to line this up, right, it has to bind it in such a way, sorry, I lost my grip, that it's a 9-23-7 paired up with a 9-12-7, right? It's a different size. And that's the only way that the oven mitt can fit around there and grab it. But in grabbing it this way, what has it done? It has actually brought this chosen red block close to this chosen green block. Anything in between, right, on that chromosome, in this case, there's a red block, it gets cut off, and that goes away as a loop, floats away. But what you've successfully done is you've now brought together one fragment with another fragment, right? And so then you can read this genetic sequence across it as one meaningful message that can be translated into a chain of amino acids that gives you your finished chain of your immunoglobulin. Okay. This always works better in person because then I get like volunteers to hold this in front of the class, and it's a lot more fun. I've managed to like wrap this around my legs now. I'm gonna like trip when I get up. Okay. Anyhow, let's go back to sharing this here. Okay. So that's what's going on here. So the oven mitt is the RAG protein complex, and because of the shape of it, right, it can only successfully grab onto two parts of the rope if those two parts are those RSS sequences, right? Because there's the orange part that's recognized by the oven mitt, and there's the purple part that's recognized by the oven mitt, but they have to be spaced differently, right? A 9-12-7 RSS sequence will only be able to be held onto at the same time as a 7-23-9 sequence. And that provides the key to keeping all of these things straight. It's kind of remarkable, right? That's what happens. All right, so just by VDJ recombination alone, by choosing from the 30 or so red blocks, the 6 or so yellow blocks, and a few green blocks, that alone means you can make 295 different light chains just by the probability of choosing different fragments. You can make over 5,000 different heavy chains, because remember, the light chains only involve a red V block and a yellow J block, whereas the heavy chains have that extra green D block. So that adds to increased, numbers of different options that you could have, right? So that alone, though, right? So that's a few thousand different variations. That's not nearly hundreds of thousands like I've been talking about, right? So the rest of it comes from things like junctional diversity. So let's move on and take a look at that. Remember, I said once the rope has been cut, you would have two free ends of rope, okay? So in other words, your DNA is cut so that the extra loop of DNA floats away because the oven mitt has chosen these two sites to bring together. You obviously have to join the DNA up again, otherwise you're in trouble, right? Because your chromosome has now been split and you'd have like two halves of the chromosome floating around. So right away, you have enzymes that go to work in joining these ends. They're joined through a process known as non-homologous end joining. What does that mean? Non-homologous means they're different, right? These two ends of the DNA were never meant to originally be together, right? Because they came from different stretches that were then brought together, and then everything else in the middle was just cut out and released, right? So you got to join these ends together. And the way it does it is that, okay, let's say here you have a D block that had one of these RSS sequences, the heptamer, the 7 base pair sequence, which I guess, yeah, even in my rope, it was orange colored, right? And then the other side, you have a J block that has the heptamer next to it, right? And this was then cut, and these two ends have to be joined together. So you have an enzyme that cuts out the heptamer, leaving whatever is left. In this case, on this one, you have a C and a G. Here you have a T and an A, right? And because the ends, the C is complementary to a G, it will actually automatically just connect like this to form like a little hairpin, right? This is a double strand of DNA, right? So the T and C on this side of the strand is complementary to the A and G on the other side. And now the ends have just kind of been connected to each other, right? So the RAG complex has other proteins that will go ahead and snip one of these fragments. So where these black arrows are, it'll snip one of these strands. So now this A and the G are cut here, and the A and G can kind of flip out, right? And it's attached to the C and the T on the other side. So you get this kind of an end sticking out. Likewise, on this strand here, the enzyme would cut maybe right here. So the A and the T would then flip out, right? So now it's sticking out as part of this. So you have these overhangs, right? Where the double-stranded DNA ends up just being one strand and it kind of overhangs. You then have enzymes that come in and start randomly adding nucleotides to the ends. Because remember, like this sequence, this TCGA, there's nothing to follow as a template, right? So the addition of additional nucleotides is random. So you can see that's what these colored things are, CTCA, right? And they start randomly adding. At a certain point, move up to here, you will get enough of them that by chance are complementary to each other. So the T has been added, and an A has been added over here, and a C and a G. You begin to get the start of these two strands coming together. And when that happens, right, the extra ones that were sort of garbage are lost. And then you have polymerases that fill in the rest of the sequence. Importantly, this little stretch of DNA, because it was randomly put together, right, was not part of the genome that you were born with, right? And yet it's part of the coding sequence that goes from D to J. Right? So that is junctional diversity. And that raises, because of the random nature of the DNA bases that are used in there, that raises the different number of shapes of IG that you can make up to 3 times 10 to the 7 in variation. Right. And then the very last thing is simply, this is happening when you're making heavy chain. This is happening when the B cell is also making the light chains. And then, of course, the heavy chain and the light chains have to come together. So all the possible heavy chains that you could make multiplied by all the possible light chains that you can make, right? When you combine those together, that's what raises the diversity of these shapes up to 10 to the 12, which is pretty phenomenal, right? Pretty phenomenal. Okay. Okay, so we're out of time for today. There's 2 extra slides, but I'll just start on Monday by finishing up on those 2 slides, okay? Because I know some of you have to go to lab, so I don't want to keep you too long. So let's go ahead and stop the recording here.